on suspension culture, production of secondary metabolites, bioreactors, and scale-up of secondary metabolites. First, we should learn about suspension culture. What is suspension culture? It is a type of culture in which plant cells or tissues grown in the liquid medium. It helps to produce the suspension of single cells or the clumps of cells. Cell suspension culture is the primary route for studying the secondary metabolism of the plants. The cell suspension culture requires optimization of cell line and cultivation media and bioreactor system. Next, we should study about types of suspension culture. There are mainly two types of cell suspension culture. One is batch culture, another one is continuous culture. Each of these culture has its own advantage and all types are being used in practice. Next one is batch culture. It is a type of suspension culture where the cell material is grown in a finite volume in the agitated liquid medium. These cultures are maintained continuously by subculturing. Batch cultures are the most commonly maintained in conical flasks because it is incubated on orbital platform shakers at the speed of 80 to 120 rpm. It is a closed system. In this system, there is no addition or the removal of the nutrients or the waste product during the period of incubation. This culture is further divided into four types: slow rotating culture and shaker culture, spinning culture, and spiral culture. The slow rotating culture means the cells are grown in the liquid medium, that is slowly and gently agitated in the liquid at 1 to 2 rpm. And the shaker culture means the cells are suspended in the liquid medium within the flask, which is then replaced on the shaker or the orbital to oscillate continuously. And the third type is spinning culture. The cells are placed in a spinning flask to gently rotate the cell suspension in liquid media to ensure the proper mixing and aeration. Fourth type is spiral culture. Culturing the cells in a closed vessel where the magnetic stirrer and the air bubbles agitate the medium to keep cells dispersed and supplied with nutrients. Second one is continuous culture. In continuous culture, the liquid medium is replaced continuously with the fresh liquid medium to stabilize the physiological state and to stabilize the physiological conditions of the growing cells. In this system, the nutrient depletion does not occur because of the continuous flow of the nutrients, and the cells always remain in the growth phase. The continuous culture is further divided into two types. One is the closed type, another one is the open type. In closed type, both the user medium is replaced with the fresh medium, and the cells are mechanically retrieved from the medium, and again it is sent or added back to the medium. Next, in the open type, in this type, both the user media and the cells are replaced with the fresh medium, thus maintaining the culture at constant and submaximal growth phase. Next, advantages and the disadvantages of suspension culture. First, we should learn about advantages of the suspension culture. It helps for the production of secondary metabolites, and it helps for the production of vaccines for the research purpose. It helps to produce the monoclonal antibodies, recombinant proteins, and enzymes. It is used in the germ plasma conservation. The germ plasma conservation means preserving the genetic stability or the hereditary material of the plant for the future to improve the crops. This is germ plasma conservation. And this system can be scaled for large-scale production of the cells. And the whole plantlet can be grown by the single explant. This system is capable of contributing significant information about cell physiology, morphology, metabolism, and metabolic events. Next, we should learn about disadvantages of the suspension culture. The productivity of the suspension culture decreases over extended subculture period. Slow growth and low productivity. If the cell grows in the cell is grown in the cell growth and low productivity of plant cells. Cells may get damaged by shear conditions. Production of secondary metabolites. There are no secondary metabolites. Secondary metabolites are the small organic compounds produced by the plants, which are not essential for the growth, development, and the reproduction. They may include pharmaceuticals, flavors, food additives, or the livestock, and the alcohol, alkaloids, phenolics, and terpenoids. The secondary metabolites are not directly involved in the metabolism process, but they are involved in the defense mechanism. The plant hormones, which are secondary metabolites, are often used to regulate the metabolism of the cells within the plant and oversees the overall development of the plant. And the function of the secondary metabolites is it protects the plant against the herbivores and microbial pathogens, and it serves as a attractant for the pollination and seed dispersing animals like honeybee. And production of secondary metabolites. The production of secondary metabolites involves three techniques by plant tissue culture. First one is cell suspension culture, second one is hairy root culture, and third one is immobilized cell culture. First one is cell suspension culture. The cell suspension culture is developed by, it is a vital technique in the plant biotechnology. It is used to cultivate the plant cells in a liquid nutrient medium. The cell suspension culture is developed by transferring a small amount of callus into the liquid medium under suitable conditions of aeration, agitation, light, temperature, and other physical factors. The agitation and suspension incubate on the rotatory shaker at 1000 rpm for agitation under the dark in the growth room. First, we should select the healthy mother plant, and then we should extract the explant from that mother plant, and then we should surface sterilize that explant. Then we produce a callus by using the solid medium. Then it is transferred to the liquid medium, the liquid broth medium. There, through that we can produce the secondary metabolites. In our syllabus, there are two secondary metabolites are given. One is saffron, another one is the quercetin. The saffron, you know, it is the most expensive spice in the world. The saffron, it is extracted from the flower Crocus sativus, and it belongs to the family Iridaceae, and it is found in India, Spain, France, and Greece. The saffron is a red color stigma present in the large flower, lavender color flower. It is a dried stigma. And the production of the saffron through suspension culture. First, we should select the corm. You know, corm is a perennial bulb-like structure present underground of the flower. This is the corm, and we should remove the outer coat of the corm. Once we remove the outer coat of the corm, it is surface sterilized with some of the chemicals like 320 or sodium hydrogen peroxide or any other chemicals. And then through this form you can develop a callus in the solid medium. In solid medium the agar is present, through that we can develop a callus. This callus is cut into small pieces and it is then transferred into the liquid medium. Through this liquid medium we can develop a new plants of the saffron or the secondary metabolites. And advantages of saffron. It improves the digestion and it boosts the pregnancy health. It enhances the immunity and energy levels. It combats anemia and reduces hair fall. It makes the skin healthy and radiant. It improves memory and nerve functions. And it cures asthma and it alleviates diabetes and suppresses cancer cells growth and tumor formation. It fights against the cardiovascular diseases and neurological diseases. Suppress the cancer cells growth and tumor formation. It is a powerful antioxidant widely used in the cosmetics. It treats the insomnia, depression and anxiety. It protects the kidney against toxic agents. Next we should learn about the capsicum. The capsicum is extracted from the capsicum bacatum. It is an active component of chili pepper. It belongs to the family Solanaceae. It is found in America, India, Japan, Mexico and Africa. The capsaicin are most widely used in the medical field. When we apply it on the skin, it creates a burning sensation. And it is a powerful antioxidant against the cancer. And the production of the capsaicin, first we should select a explant and the surface sterilize the explant and produces a callus from the solid medium and transfer this callus into the liquid suspension medium or liquid nutrient medium. Then it is transferred to the bioreactor to produce in the large scale or in the large amount. Advantages of the capsaicin. It is having an antioxidant activity and antimicrobial activity. and it protects against the um, it is a gastroprotective effect and helps for the cancer prevention and cardiovascular benefits, anti-obesity effect, inflammation and curing. The second type of culture is hairy root culture. Hairy root culture is also known as transformed root culture. It is a type of plant tissue culture used to produce a high volume of secondary metabolites or recombinant proteins in vitro. The Agrobacterium rhizogenus causes a hairy root disease in the plant. The neoplastic roots. The neoplastic means tumor. The roots having a tumor, that is known as the neoplastic roots. The neoplastic roots produced by the Agrobacterium rhizogenes infection are characterized by the high growth and genetic stability and high growth in the hormone-free medium. Hairy roots are induced by genetic insertion of tDNA. tDNA means transfer DNA. tDNA of RI plasmid. RI plasmid means root-inducing plasmid. RI plasmid lodged in the Agrobacterium rhizogenus into the wounded tissue, causing the growth of very fine adventitious roots. These are the very fine adventitious roots, which are produced by the infection of Agrobacterium rhizogenes. We know that when the plants are wounded or damaged, it produces one chemical. The chemical name is acetylcysteine. The genes of the bacteria helps in the attachment of the plant cells. And the weir regions, we know some vector, the Agrobacterium rhizogenes, it is having one weir region. This region helps for the transfer of tDNA into the plant cell. The RI plasmid has a left transfer tDNA region and the right transfer tDNA region and can induce the root formation in the plant. Third one is immobilized cell culture. The plant cell membrane can be made immobilized or immovable. The plant are immobilized by physical entrapment. The common entrapment methods are entrapment in the gels and the entrapment of the cells of the nets and the entrapment of cells in the hollow fiber membranes. This is the entrapment of the cells in the gels and this is entrapment of cells in the hollow membrane, entrapment of cells in nets or the folds. The surface immobilized plant cell technique retains the cells and allows them to grow at a higher rate. It improves the better cell-to-cell contact and cells are protected from the high liquid shear stress. All this helps in the maximal production of the secondary metabolites. Next we should learn about bioreactors. Bioreactors are the specialized vessels or the reserves which provide a controlled environment for the cells, microorganisms or the enzymes to grow and produce a desired product. These devices provide optimal conditions such as temperature, pH and nutrient levels, agitation and etc. to ensure the biological processes occurs efficiently and at a high rate. The bioreactor is involved in the suspension culture. There are mainly three types of bioreactor. One is sterile tank bioreactor, another one is aerial bioreactor and another one is wave mixed bioreactor. First one is sterile tank bioreactor. Sterile tank bioreactor is commonly used for cultivating the plant tissues or organs under controlled conditions to enhance the production of secondary metabolites. And this is the agitation system. This agitation system ensures the uniform mixing and oxygen distribution. This is aeration control. It maintains the optimal oxygen level and the cell metabolism. Temperature and the pH regulator. It regulates the optimum pH and the optimum temperature. And sterile condition. It maintains the sterile environment for the long culture periods. Advantages. It is suitable for long-scale production and its controlled environment allows precise manipulation of growth factor. It enhances the metabolite yield. It supports the suspension culture. Second one is airlift bioreactor. The airlift bioreactor is a type of pneumatic bioreactor. The pneumatic bioreactor means it uses the compressed gas in a particular region. to create to agitate the liquid or the cells in the bioreactor, or the proper agitation of the cells in the suspension culture that uses the air or gas to circulate the culture medium. It is typically consists of riser and the down cover tubes. This is the down cover tube and this is the riser. This creates a loop for the fluid circulation. It creates a one loop that is for the circulation of the fluid. And the gas spranger. The gas spranger induces the air at the bottom of the riser, and the internal and the external loop design. This loop design determines the flow path of the medium and low shear environment. It is ideal for delicate plants. And advantages: due to the low shear stress, it protects the fragile plants from the damage. Ventilation promotes the cell growth and metabolite production. No mechanical stirrer, reducing the operational cost. Third one is wave mixed bioreactor. Wave mixed bioreactor, also known as wave bioreactor or rocking bioreactor, are increasingly used for producing the secondary metabolites in plant cell suspension culture due to their gentle mixing, scalability, and sterile operation. A single-use flexible bag is partially filled with the culture medium. This is a single-use flexible bag. It is partially filled with the medium, and it is placed in the rocking platform. It creates a wave-like motion. This is the wave-like motion created in this bag. And the surface aeration from the headspace provides the oxygen transfer. This design eliminates the mechanical agitation and reduces the shear stress and contaminated plants. Advantages of the wave mixed bioreactor or low shear environment: it protects the fragile plants, fragile cells and aggregates. Efficient mixing and gas exchange: it promotes the uniform growth and metabolite synthesis. Scalable volumes: through this bioreactor, we can scale up 0.1 liter to 500 liter, suitable for large industrial scale. Closed system minimizes the contamination and simplifies the cleaning. Scale up of secondary metabolites. Scaling up of the secondary metabolites in the plant tissue culture involves the transition from the large scale production to industrial scale production by maintaining the yield, quality and the cost effectiveness. First, we should select the another technique is by genetically engineering the cells. We can synthesize or we can increase the production of the secondary metabolites. Optimization of the culture condition by providing optimum pH and providing optimum temperature or sufficient nutrients. We can scale up the production of the secondary metabolites. By using suitable bioreactor, we can scale up the production of the secondary metabolites. We already learned that airless bioreactor and the wave mixed bioreactor and the stable tank bioreactor. The airless bioreactor is ideal for the low shear environment. It means it applies only less pressure on the cells or the plants cells. And the wave mixed bioreactor, it is great for gentle mixing and the stability. The stable tank bioreactor, it is suitable for robust cell lines and high density culture. Elicitation technique: by adding elicitors means inducers. The elicitors like methyl jasmonate and the salicylic acid, we can scale up or we can increase the production of the secondary metabolites. By reducing the stress conditions like UV light, osmotic stress to enhance the yields. The process monitoring by real-time sensors track biomass, dissolved oxygen, pH and metabolite concentration. By taking feedback control system to maintain the optimal condition. Through this, we can increase the production of the secondary metabolites.